to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 11th episode of the Weekly Bioanalysis. My name is John Perkins and I'm a Senior Scientific Advisor for all things related to LC-MS technologies. I've been with KCAS for almost 9 months, but I've been in Bioanalytical for close to 25 years. I am here with my co-host, Dominic Guarino.
Hello, my name is Dominic Guarino, and I'm the Senior Scientific Advisor for all things related to ligand binding assays and biomarkers. I've been with KCS for about 7 years. I've been in the CRO space or the drug development space for about 25 total years.
Today's podcast, as always, is brought to you by KCAS, Kansas City Analytical Services. KCS is a bioanalytical laboratory located in Kansas City, serving the pharmaceutical and biotech industries for over 40 years now. Dom and I are senior scientific advisors at KCS, as we previously mentioned, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCS's services. We are thrilled to have you listening again. As mentioned, this will be our 11th week recording the podcast, so if you're interested in other episodes, can be found at kcsbio.com. As a reminder, you can follow us on Twitter at The Weekly Bio and at KCS Bio. As we mentioned before, we started this whole thing because of social distancing getting in the way of normal communication. Even today, Dom and I are in completely different places. I'm in upstate New York, Dom is in Kansas City, and our producers, Jeremy and Erin, are in different places around the Midwest. This weekly podcast gives the chance to connect with all of you, and we hope it's a chance for you to get to know us too. And even more, it's a chance for you to get to know KCS and the services we provide. As always, a review of the latest news and resources, and then a focus on topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. Then we'll wrap up and give you a little teaser for next week, letting you know what is coming in the future. So again, we're thrilled to have you here, and we're looking forward to a fun episode today. To kick us off, Dom is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, John, thanks. We got a jam-packed show today, as always, and we'll first start with our news and resources. Then we'll go into our main portion of the podcast, which is where does bioanalytical fit in the drug development process with a focus on clinical studies. Got a lot to talk about there. Next, we'll move on to any feedback and questions that we got from our audience. And then we'll end with a little bit of a wrap up and talk about what we're going to do in next week's podcast as we continue with our series. Although without sharing too much, we'll talk about what we're going to do as a slight turn from the norm with 2 of 4 series. So John, with that, we are again in part 2 of 4 in our series on how does bioanalytical fit in the drug development process. And let's just jump right into the news and notes. The first topic related to COVID is inhaled dexamethasone. Dexamethasone burst on the scene a couple weeks ago. It's a potent steroid. And now they've made an inhaled version of it. I'm sure at some place they had that on a shelf, but makes total sense to me. For treatment of late-stage COVID patients, it seems to be at the site of where the infection is most active in the respiratory system, in the lungs. So why not make an inhaled version of it? Really nice story there to see that they're continue to make progress towards knocking the death rate down for COVID, despite a big spike in COVID outbreaks, but certainly less and less on the death side, John.
Yep, certainly it's interesting with the dexamethasone, the recovery trial in the UK that had announced it was an effective medication, published data and really emphasised that dexamethasone is really there for use in late stage patients and is of little to no use in early to moderate. So it's there in the armory to help us, but really for very, very sick people. But great to have it. I'm sure if you've got people in inhalers and they're introducing drugs via that route, then that to have the inhaled dexamethasone is a good thing to have available.
Yeah, definitely. And again, I think as we're seeing these spikes, what did I see on the news this morning? The highest level of COVID in the US since like sometime in April, but the death rate really stays low. And I'm firmly a believer that we're treating it better. We're probably identifying it quicker, and all of that will help with keeping that death rate low, which is really what it's about. Although no one likes to get sick, and there's still some caution around outbreaks. Certainly, The next item on the news list here was we had an outbreak here in Kansas City at a Chick-fil-A. 10 of 80 people tested positive. I think we're going to see more of this. I think it's happening more. And it's something where it's just mentioned on the local news and then we on to the next topic. But those are some concerning things. One person exhibited some mild symptoms, and then I happen to know somebody that worked there, a young man worked there that I know, and he got tested for it. By the way, he says getting that nasal swab, they stick that big 9-inch sort of cotton swab up your nose. He said it hurt and didn't like it, but it is what it is. But the other 9 people exhibited no symptoms, and I think that's really probably what's happening. I think there's a lot of somebody might exhibit a symptom and then they tested a whole bunch of people and finding out they're positive. Certainly a lot of outbreaks in sports and stuff like that. So kind of cautious about things, and it's kind of frightening to see these outbreaks, but let's hope again the death rate stays low.
Yeah, I think there's a number of things at play, aren't there? I think with the rising cases but lowering death rates, I suspect some of it is, like you say, the care's getting better, catching people earlier. But it also might be the fact that we have medical supplies in place to deal with them. Certainly in the early days in New York, a massive flurry of inpatients, etc., which greatly strained the resources there. And I know I mentioned before that people were sent down from my area to help out in the city. But right now, I think there's the capacity to take patients on if they need to be taken into intensive care. Although Texas, I think a couple of locations are saying that they're close to maxing out on available bed space, etc. Um,
Yeah, Florida too. Florida's having that issue, I think. Yeah, the bed spaces are getting pretty full there. Sorry, go ahead, John.
This troubling thing for anyone watching this is the proportion of people who are asymptomatic got no idea who you could possibly catch it on, because there's a significant proportion of people who aren't showing any symptoms at all. Outbreaks are popping up in sort of really odd places. I don't know if you saw with the tennis, there was a tennis tournament, I think, Croatia, run by Novak Djokovic, who's a Serbian, and he was a big part of it. And both countries bought into what he was trying to do, and then it all sort of unraveled as the week went on, as more and more players tested positive, including Djokovic himself. It all indicates you can't let your guard down. And as we've said so many times, hand washing, social distancing, face masks, do that, and you're going to help keep the transmission down. And you just have to be cautious and maintain those controls. And actually, another thing that I was looking at earlier, the UK is really pushing to drop social distancing from the 2 meter to 1 meter. Don't know what the basis is, other than they really want to obviously get the economy moving again. But there's a clip of a British scientist saying that the dropping from 2 meter to 1 meter actually increases the likelihood of transmission, increases the rate, I think, by 10 to 30%. Please listen to the scientists and take precautions in line with what they're recommending. And recommendations will change as we learn more about it, because obviously we talked about face masks in the early days. People were saying, face masks aren't going to buy you much, but obviously the more information that comes out, the more it says that they are a critical part of keeping transmission down.
Yeah, no, I couldn't agree more. And you sent that really scary photo of the beach in, I think it was South England, where it was just slam packed. I mean, it looked like, summer of 95. It looked like COVID had never happened on that beach. And forget 1 meter, John, that looked like there was less than a centimeter between everybody on that beach. And that was quite disturbing. It's definitely an economical thing to want to move from 2 meters down to 1. And I still think if even keeping a meter will help, but clearly, like you said, 2 meters is definitely the range where you can really prevent aerosols from being discharged from a sick person. So here in Kansas City, it's definitely much more relaxed. I'd say 1 in 10 is wearing a mask. But, other than that, Chick-fil-A, I did look on the CDC website. Our rate has not skyrocketed. Our death rate has stayed right at about 160. Sometimes it goes up and then it goes back down. It's really weird how they must miscalculate or misdiagnose COVID deaths. But yeah, here, I think we're in a much better state than, certainly Florida and Arizona and places like that. But again, maybe it's some testing, but certainly there's got to be some lax in social distancing, as well as even just, people going to nightclubs and all that stuff that's really, I think, causing some major hotspots in our country.
Yeah, I'm happy that it has maintained low where you are, but ultimately what we're seeing is it doesn't take much to trigger it up. It just takes that one or two carriers and in confined space, and it can easily take off and give us another hotspot. Let's move on. You should actually introduce this one as well because this came from you.
Yeah, this was an interesting one from the UK. They launched a new national COVID-19 test validation project, which I thought this was really great. So they're trying to really streamline the validation process of each of the tests so that they can get a good handle on, who's positive and who's negative, and they're the first to launch it. I think this is a wonderful thing, and I really hope that something in the US sort of emerges that says, hey, here's the battery of tests that are validated here, and we're going to make sure that we keep a good tab on, how many there are, how many they're giving. I just think it's a wonderful approach towards trying to get an understanding of who's positive and who's negative, and, kind of really, I still don't think we have a good handle on percent positives and all that sort of stuff. So it was an interesting read.
I think this is great. And actually, when you said about hoping the US would do this too, I would hope that if the data that this group are publishing is good enough through international cooperation, it shouldn't have to be replicated in other countries, and they could sort of set a mandate as to what tests are actually useful and which ones aren't. So I think it's great. I do like it as an initiative. And kudos to the National Institute for Health Research UK, Asthma UK, and the British Lung Foundation for getting this off the ground.
One last remark. I don't know. The EU and the US have been at odds. The EMA guidelines and FDA guidelines can't agree, John. So I think you're asking a big ask there. Let's move on to the next topic. There's a strained supply chain for glass vials that could delay coronavirus vaccines. So, the vaccines are really upticking, but this was an interesting, in addition to just manufacturing issues, really makes total sense that glass vials could be at a premium. But again, just another sort of obstacle in the way of getting a vaccine out there. And I'm sure it's going to impact all vaccines if there's a glass vial supply. So just one more thing on the list of challenges towards getting a vaccine to the public.
Yeah, it just shows how it's the simple things that can derail you. I mean, we sort of focused on here with developing a vaccine, putting it in trials now. I know we discussed logistics where we're talking about pharma companies outsourcing to other partners to increase their production capacity so they got billions of doses. But it could actually be glass that brings everything to a halt because not many suppliers out there are being able to get tubes together. And I agree. I think you're right. This could have a knock-on effect to other vaccines, which is, the last thing you want. I mean, you want to have the ability to ramp up COVID-19 and still be able to administer and produce vaccines against, what are still very nasty diseases all around the globe.
Yeah, maybe those automakers, I don't think car sales have rebounded enough. Maybe they'll be making glass vials for vaccines. We got manufacturing plants that are probably going to be repurposed. And this is just another one where I'm sure they could turn out millions of them if they just shifted their focus. And it could very well be coming. And, on the vaccine front, well, maybe we'll just take a quick update there. Recently there was I forget exactly who it was, but they were predicting that we'd have a vaccine by the end of the year. One of these candidates would emerge. I'm not sold on that. I think there probably is an effective vaccine, but just getting it through a phase 3 trial and looking at gender and race and age and pediatric and all of that, it just seems like we're moving way too fast to think that that's going to happen by the end of the year. But, I hope I'm wrong, but certainly there's enough vaccines out there that I started thinking maybe I should get on a trial. But anyhow, and all kidding aside, I think that getting a vaccine by the end of the year is just maybe more propaganda, and maybe that's the right thing to do. Maybe it's to give people hope, to give people some sense of security. But really, I would be really surprised if anything was like approved before the end of the year. And even I think going into next year, I feel like we better temper our expectations around having a vaccine readily available in 2020 or even 2021.
Yeah, I definitely tend towards the cautious on this too. I don't expect it in that timing. I mean, I think that really is at the optimistic end of things. And having seen how many things have happened to knock trials back that we've all worked on, I hope it gets there sooner rather than later. But, reality is bound to intervene.
Yeah, and let's stay on the vaccine topic. And Regeneron's COVID-19 antibody cocktail cuts the risk of resistance. So this was a real interesting read about how they got 2 monoclonal antibodies using their mouse system, which is a fascinating system of how they're generating these. If you recall on a previous podcast, they had just came up with the target that they wanted in February, and now they're already into like phase 2 trials with these antibody cocktails, but it's an overlapping pair of antibodies targeting one of the proteins. They don't specify much around it, and rightfully so. It's real proprietary stuff, but that's a potent way to do it. They're looking to sort of take 2 two shots on goal in the same cocktail, and really hopefully this becomes a real potent vaccine.
Yeah, it's that approach of developing two antibodies that attack different parts of their target is hopefully there to guard against mutation and resistance. It could be very interesting.
Yeah, and then staying on COVID was the study you found in China, and I'll intro it and then you can share some details, but it looks like antibody levels in recovering patients declined pretty quickly, John, right?
So this was a small study in China where they looked at 37 symptomatic patients and 37 asymptomatic patients, found that those who had tested positive for the presence of the IgG antibody after infection, that actually rapidly declined over 90% in 2 to 3 months after infection. So it's raising concern about what is the likelihood of immunity to the disease after you've caught it once. And I know there was a couple of comments on that to say that, yes, there's other parts of the immune system could offer protection, but it's still one of those things that it's just a note of caution to sort of say, just because you've had it once, it doesn't mean that you're proof against it a second time. So that had been partly postulated as to people who tested positive or had the disease could then be applicable to go and do much more traveling. Well, this might be a little bit of a yellow flag to that.
Yeah, I mean, I would have always cautioned that mindset. I mean, the N number in the study is a little low, but I think it would hold true even if you powered that up, meaning it's not surprising that if you come in contact with an antigen once, an antibody lasts circulating anywhere from 30 to 60 days, so seeing the IgG levels decline, it's not surprising that what they didn't address was memory, both memory B and T cells. That's really where your immunity gets protection. I do feel like if you've had it once, you're almost guaranteed to have some protection when you go to get it again. I think that's true. And then, even look at how we do hepatitis or even HPV. It's a series of vaccines. So that's just how the immune system works. You have an initial inoculation and then a boost and then a second boost in the hepatitis B to ensure that your protection and your seroconvert and you've got circulating and long-lasting memory. What I really would have liked to have seen is some results flow cytometrically, and I think some of that is coming. Staying on the COVID topic, we got, I did have one of the clients that we're doing biomarkers analysis for shared a recent publication, and it's great how quickly the field is moving for biomarkers in the COVID space. And we know the inflammatory markers such as IFN gamma and IL-6 and IL-8. They're starting to see that maybe TNF alpha isn't as big a predominant of a cytokine in some of the ICU ventilator patients, which I think is a clue. There's some clue in there about what that means. But then we talked about D-dimer. That's one that is an active biomarker. And then very nicely, we're seeing things like C-reactive protein, which is part of your liver. So maybe the liver's involved. And then there's lots of respiratory markers that are coming across as being important in terms of biomarkers. And then there's a flow panel that they're eliciting along the T helper 17 pathway. So really some nice advancements. And these are all things that are really going to help treat the existing patients. And I think to some degree ensure that the vaccine's working too. I think that would be another route of ensuring that we've got protection. So with that, let's move on to our last topic. Mini organelles reveal how the coronavirus ravages the body. Really cool stuff, John.
This is one of the things that points out the value of just following random people on Twitter. I would never have found this otherwise. But this is amazing in terms of researchers grow miniature organs in the lab to then study how coronavirus ravages the body. They got these mini organs in vitro, and they acknowledge that it doesn't completely cover the interaction that would occur within a complete biological system, but still gives a chance to assess how the virus attacks things. What was interesting to me, we've talked about the route is in through the lungs, but they're actually seeing impact on the liver, kidneys, and gut. So just shows how insidious this virus is. And so where they were thinking, conditions were a secondary effect of the virus, they're actually finding that, based on these experiments, that actually it's the virus is directly causing these issues. So it gives you that, more of an ability to then figure out how can you address it. So absolutely fascinating paper.
Yeah, viruses are such fascinating things in and of themselves. They're evolutionarily, they're really coy creatures, meaning they do things to knock down your immune system. And that was one thing they're finding. Another potential biomarker is your helper cell levels and CTL levels, cytotoxic T cell levels are getting knocked down by COVID, similar to HIV. That's another potential sign of infection or at least progression of the disease as a biomarker. But back to these organelles, just such beautiful stuff they're doing. using 2D and 3D type things. And really, John, I mentioned CRP as a liver enzyme. It's not surprising to see that it's attacking the liver. And there are undoubtedly, I think, a lot of other biomarkers that are going to get unearthed. And maybe some of that will start to look at strains of it, and even just getting back to how much you actually ingest may have an impact, meaning getting only a few particles. Maybe it stays in your respiratory and you stay somewhat asymptomatic, get a huge bolus of it and it starts to get through your circulation. And next thing you know, you got a lot of organs that are triggering immune responses, and the virus starts to aggress, right? That's when it starts to grow in cells and then aggress into your body and everything. So fascinating stuff. Let's move off of COVID. The first non-COVID item was to me, just unbelievable story that Bayer had to pay $10 billion for the Roundup settlement. I'm sure they're shaking their heads over purchasing Monsanto. I think it's great for these individuals who had some sort of cancers, I guess, related to Roundup, but this hopefully will put this behind Bayer and they can move on. But 10 billion is just a massive amount.
It's incredible, isn't it? They paid 63 billion for Monsanto a couple of years ago, and then they're having to pay this out to settle lawsuits. So it's a combination of Roundup, also known as glyphosate, another herbicide dicamba, as well as water contaminated with PCBs, and this is addressing tens of thousands of lawsuits. Although it's not, I think they say it addresses 90% of lawsuits, so it's still not completely finished. But they're obviously making steps to at least make some sort of reparation. But yeah, it's astonishing.
Next, non-COVID, AstraZeneca was the first to join FDA's push for patient-recorded symptoms in cancer trials. So if you're not aware, within any sort of drug or cancer drug in particular, there are the black box, it's so-called, but that includes a series of adverse events, all sort of safety issues. Well, now AstraZeneca stepped up and heard the call from the FDA, and now any patient-reported symptom would be available to the public. This is, whether it's a runny nose or anything like that, you'll be able to go and look before you take a drug. I think this is a long time coming. We've got information highways out there, and there's no reason this shouldn't be available if you or a loved one need to take a drug.
So I'm all in favor of transparency, and it helps people be informed about the medicines that they're being administered. And we know, with particularly cancer drugs, they can be pretty nasty compounds. So I think it's great to have that. Here's information to help you give a bit of background to what you might expect taking this medication. Although I mean, I do. Obviously, we hear when you see the commercials on TV, we talk about how it's going to change your life, and then there's always the disclaimer at the end about all the side effects. I wonder if the commercials are now going to double in length because they're listing all the side effects that patients have acknowledged. So it'll be interesting to see.
There's one last item, and I think it's important to mention. That's MIT researchers developed strong yet detachable surgical tape. And so that's for when they're in doing a surgery and there's some unexpected internal bleeding, or even if they may have a nick, this particular tape that as I read it, will be able to adhere and stop that bleeding. And then they have some sort of solution they put on there and makes it very easy to remove. And I think this is, again, a great sign to see more of the research going on that's not COVID related. And we have COVID, but there's still a need and a demand for science and research. And this was just a really cool technology that I'm sure is going to help save a lot of lives.
That's fantastic, isn't it? You always think when you rip off a Band-Aid, if it's stuck, how much it hurts. If that actually is easy and it comes off easily when you want it to, that makes your life much more pleasant.
To your point, I think in time, you'll see that be the Band-Aid, right? Like, hey, in order to remove the Band-Aid, put a few drops of water on it, comes right off. So great stuff. I do want to mention some investments, John. I like this. Merck penned a $500 million deal with Humanity, and that's around some neurodegenerative hopefuls. That's two particular drugs. Merck was a little late to the game on COVID, but good to see them getting in the investment. And it's always just good to see investments. And then I'll quickly mention two, and that's both a company called Simcha raised $25 million for IL-18 research, and then Sanofi purchased Synthorax for $2.5 billion. Big delta there, right? To take over, gain and control of IL-2 cancer drugs. Well, IL-18 and IL-2 are linked. They share a receptor. These are very potent interleukins. Back in the 90s when they were giving IL-2 to people, if you could live through that cytokine storm, you tended to cure the cancer, although a lot of people crashed and succumbed to the actual cytokine storm. But, this is, again, people are chasing that. We're working on a couple IL-18 drugs. We're working on a few IL-2 drugs. I know the IL-2 in our instance, a lot of them are pegylating it, but really cool stuff. And they've been around a while, and let's hope that they can continue to make some advancements so that they can get these into mainstream therapies.
This was really interesting looking at the Synthorex approach where, obviously, like you say, there's issues with recombinant IL-2s, but they think by inserting in a novel amino acid at one position is enough to change the character to get away from the downsides of IL-2s, and then they have other candidates in the pipeline beyond that.
And with that, we'll wrap up our news and notes, and we'll move on to the main portion of our podcast. This is our 11th podcast. Look at that, John. That's wonderful, right? This is part 2 of 4 in where does bioanalytical fit in the drug development process? If you recall, last week we talked about preclinical and animal studies, and this week we're going to talk about clinical studies.
Yep. So before we get fully into clinical, I'd like to take a step back to preclinical, because one of the things that we didn't touch on is the way that preclinical studies are run in a GLP setting. They actually have a single point of control, and that's the study director who's assigned at the tox lab. So the animal facility, they assign a study director, and they're the center point of control for the entire study. So all information funnels through them. If we have any issues that we observe in the bioanalytical, we have to let the study director know what we've seen. So if we have a deviation, they have to be informed so that everything funnels through that single person. And then they report to the pharma company that they're working with. When it comes to clinical, it's a lot looser. We don't have that same flow of information. There are people that will lead the trials, but we don't quite see that this person is really the be all and end all within this study. I meant to touch on it last week. We'd finished the podcast and I was thinking about it and thought it would be just good to mention this. And also it would be good in the longer term to actually have someone who's acted in that study director role to talk about animal studies from their perspective, as opposed to us talking about it from a bioanalytical perspective.
Yeah, that'd be fascinating. But John, it was when you brought it up, it was really one of those like, gee, I've been doing this a long time, and I never really thought about how the principal investigator on the preclinical setting is they hold the conduct of the study, and it's really something where, like you said, documented and really at the forefront. And then you get into the clinic and it's absent. The method is still validated, but it's really kind of on the phase 1 unit, I guess. And they might have somebody who's a dedicated person, but when you get into multi-sites, there's no one PI in that setting. Yeah.
I think that's the difference. The change in approach is very different from study to study. Even for a compound or a molecule that's being developed, you still see difference in approaches in terms of how things are done. Obviously, early phases, at least for many drugs outside oncology, you're talking volunteers just to get an indication of what the drug's doing in the body. And then as you move into later phases, you're going out to, in some cases, specialized clinics for patient populations. You're actually looking at different things. So from our perspective, in the early studies, we're looking at pharmacokinetics in terms of what the drug's doing in the body. You'll end up doing other trials like drug-drug interaction studies, which will have been based on some of the early discovery work where you may have done early just interaction tests, early metabolism screens. And so you got an idea of where this drug may impact metabolism and what other drugs it's likely to interact with. So then we have to do the formal studies to determine if that's a major problem or not. And this is the basis of some of the black box labeling that we talked about a few minutes ago. So from my perspective, for small molecule, what we tend to see is that we're very involved in phase 1, where it's the PK data is the important piece. As we move forward to phase 2, phase 3, which are the big studies, and we always say when we're getting involved in clinical trials as a small molecule provider, yeah, we really want your phase 3 samples. But what we find is, even though we do see drugs go to phase 3, our role actually diminishes unless there's population PK, which are the much more sporadic sampling. So, for instance, the biggest study I ever worked on was a phase 3, but it was a cardiovascular drug. It was probably 6 to 8 samples a patient, but that was over 2 or 3 years. Very, very different approach from the early studies. But then what we see is, and this is where you more come in, is that the biomarkers and the pharmacodynamics become much more important in the late phase studies. So where our role is in terms of assessing PK is diminishing, then it's much more what are these drugs causing in patients, etc.
Yeah, John, expanding upon that last line of thought, definitely as you get into later phases, the biomarkers and the PD markers really start to kick in. PD wise, that's where the flow cytometry comes in. And those are a lot of patients. They don't sample quite as much as PK, but still it's a tremendous amount of samples over a couple years. And we really support several phase 3 trials. We're very good at managing it. And that's where the clinical kitting that we talked about in a couple previous podcasts, that really helps manage that. So, but to take a small step back into how clinical trials for PK, and it's interesting, John, as I was sitting here, it gets quite different. Preclinically, it's pretty similar between small molecules and large molecules, with the exception of there's the ADA component that you need to look at in the preclinical setting. But as soon as you get into the clinic, I started thinking about it. The PK doesn't fade away quite as fast. Now, there is less sampling in phase 3, but I think it might be just due to immuno-oncology drugs. They don't have a big N number like some of the small molecules might in some of their trials. So I think that's why it's maintained in phase 3. And we don't see quite the drop off as you do in a small molecule, but for sure, they don't do as many samples. It's not going to be 8 to 12 samples in a patient in a phase 3. But I do think maybe it's partially the safety of it. The whole immunogenicity piece plays a part in it, but it is not the major drop off like you see in the small molecule side. And then,
I would think the immunogenicity is a big part of it. I think for small molecule, by that stage, you've got a very good handle on what the molecule is doing and metabolites, because they're going to be part of the issue too. But you're really going to know what the drug and any metabolite is going to be doing in the body.
Yeah, and I think in general, small molecules behave similarly in each individual, right? Maybe weight plays a little bit of a part in it, but I don't think like whatever your HLA types are and some of your immune system, they don't play as big a role. I think that's why you see more of it in perhaps the biologic side, because those things do play a major role, right? Because your body can respond to it as foreign and develop an antibody. So therefore, diversity around race, age, and gender are all going to play a bigger impact, and I think makes for different PK profiles, although monoclonal antibodies in general have a pretty standard profile. Really a fascinating topic here, John, to think about how you see this divergence. As preclinically, it's pretty clear, but when you get into the clinic, it definitely can veer. And I think, in the immuno-oncology space, it sticks around. Now, as you get into different, and this I think is the other part, you're frequently trying to treat with other different, you might try and treat lung cancer and you might try and treat colon cancer. Well, then I think you continue with the PK in each of those different sort of disease states, which is frequently what happens. Things like ADCs. Now, ADC, you see a lot of PK work on the small molecule though, right, John?
I was just thinking, if I look back at the drugs where we've supported it all the way through the development process. So you start with small molecule. We start off with single ascending dose, multiple ascending dose. You're basically looking at the dose proportionality in terms of accumulation in the body. There's also seeing if there's any adverse happens in the course of that. But it's always looking at populations rather than, like you say, specific people. I know that we've done, liver impairment studies, renal impairment, people who are impaired. So a group of, like I say, a population to make sure that they are not affected by their condition and taking that drug. Like I said, drug interactions. It's always looking at a group of people. That might be the difference. Yeah, like I say, more predictable for a population. I can't think of many studies we've done that were race related, although occasionally I would see something where it's a trial with, say, an Asian population, because that's where the drug's intended to go. It's actually down more to where it's going to be applied rather than specifically concern about it on anything other than a commercial basis. And then pediatrics are a big part of that too.
Yeah, I mean, for the PK, it's not as big a focus. Like we don't, in the bioanalytical space specifically, there's no, hey, let's look at female matrix and male matrix. But boy, in the ADA space, you sure do. That has a huge, that's something that for many of the drugs, I'd say about half, we just go ahead and, I don't know how much you know about how we set cut points for immunogenicity assays or ADA assays, but we typically look at 50 lots of individual normal donor matrix. And of those 50, we frequently get a 25-25 split of male and female, and we try to age match based off of what they plan to treat age-wise. So that becomes an important point in terms of, some of the big differences between small and large molecules. I mean, not only is it ADA, but you really do start to look at age and gender and things like that. And sometimes I can think of a handful where we've looked at different, Caucasian, Black, maybe even Native American and so forth to determine things. And those are usually really related to T-cell specific responses or drugs that are impacting your T cells specifically, where most monoclonal antibodies aren't doing that type of reaction. Although I shouldn't say that, because more and more bispecific antibodies are starting to look at those types of things, meaning checkpoint inhibitor and then trying to target a T cell to activate it. So that's a real potent way to look at things, and we're doing more and more of that. So then I think another thing that is slightly different is we don't have drug-drug interactions, but what we do have is co-meds. And I mentioned checkpoint inhibitors. Those are PD-L1 antibodies and things like that. Those are, almost every immuno-oncology trial has an arm now that's looking at PD-L1 antibody therapeutics. So we're doing more and more of that. We're on the verge of hopefully offering up some off-the-shelf methods for that, and some other legacy drugs. There's one called cetuximab and rituximab and Erbitux. Those are all things that we frequently have to look at as co-meds. So slightly different than, say, a drug-drug interaction where, maybe you're taking aspirin, and I'm simplifying it in your world. But in our world, we just have to look at, hey, what's our PK look like when there's not only the drug on board, that's how we describe it, the PK and ADA when drug's present, for ADA, PK, obviously we're measuring the drug, but then what's the impact of having the other monoclonal antibody in your bioanalytical method? And that becomes important because you got a lot of FC portion of it, which is sort of the anchor portion of it, can get quite complex because you're introducing, typically you have a capture antibody that's got an FC. You're trying to detect a monoclonal antibody that's got an FC. You're coming back with some sort of detection antibody, and that's got the FC portion of it. Now you spike in another monoclonal with FC. You can get some real challenges there bioanalytically that have to be dealt with. So interesting stuff, John.
We do see co-meds as well. So, we've supported in oncology where it's been a small molecule administered with an antibody. Your method is looking at the small, but you're still going to test for interference from the antibody in terms of the LCMS. There are drug cocktails administered, and certainly part of, because we got that additional selectivity in terms of retention time and transitions on the mass spec, we can largely eliminate interference from the assay, but you still want to check in terms of that there is no interference. There's been a lot of controversy about stability of your analyte in the presence of co-meds, and is there any effect on that? I've never seen any effect in any of the validations that I've run, but it's still a question that's being asked. That interference check can be essential. I remember doing a study of a drug where actually it was co-administered with quinidine, and so again it was a drug-drug interaction in this case. But when we ran the LCMS assay, the quinidine co-eluted with the analyte and massively suppressed the signal. So you could see this huge dip. If you infused the drug and then injected quinidine, when it got to the point of elution, there's a huge dip in that infusion, and it probably reduced signal by about 90%. But we had a stable label internal standard, and actually, as far as we could tell, it didn't materially affect the quantitation, so we still went ahead with the assay as it stood rather than redeveloping it. But you've always got to be thinking about what's being co-administered, and could it potentially impact what you're doing so that you can guarantee your data are solid.
Yeah, and John, I can't think of an instance in the 15 years I've been doing this in the CRO space, and even in the 25, where we had to look at a small molecule with our bioassay for a large molecule that wasn't an ADC. Let me caveat that, right? So if it's an ADC, it's just naturally going to be in there and you look at it. But I can't think of certainly not in a validation. Maybe it's something you cover in development, right? So long as you have some data that says it's not interfering or perturbing your assay, then I think you can cover yourself that way.
That's pretty much where we came from, because this was an oncology drug where initial development validation was for that oncology drug, and I think metabolites as well. And it just so happened in one of the many studies they co-administered an antibody drug as well. And actually, I've seen maybe 2 or 3 instances of that, and there was always debate with the customer whether we should do an interference check or not, because how would something that's hundreds of thousands molecular weight interfere with something that was 300 or 400? And I'm not sure whether they were right or wrong on that one, but we still ran an interference check to show that the assay was good, because it meant they had the data to say that it wasn't it wasn't affected by the presence of this antibody. I mean, it's unlikely to get through the extraction, but you still want to show that. And actually, a point about the reason for like drug interactions studies, etc. That we talked about the black box, and this is what actually feeds some of the black box information. Many of us are on statins, and one of the recommendations for statins is not to take grapefruit juice with particularly atorvastatin. I think some of the others, and that's because grapefruit juice actually interferes with the metabolism of atorvastatin. Being formal, drug interaction studies to show the effect of grapefruit juice on atorvastatin metabolism, and I think it slows it up so you get too much accumulation of atorvastatin metabolites, and I think that's the concern. But these are the value of the studies, and then that's what they're leading to in terms of the information that's supplied to the patient.
I'm sure there's a fascinating story around the history there, and I'll share what I think is a good historical story for why the whole field of immunogenicity came about. I mean, when I was doing autologous T cell therapies, we were using a mouse monoclonal antibody, and we were looking at human anti-mouse antibody responses. So they were called HAMA responses, and then it evolved into human anti-human antibodies, AHA, and so you still hear that term sometimes. And then there's human anti-chimeric, HACAs, and then there's human anti-therapeutic antibodies. That's now all been dubbed immunogenicity, and the main driver for it was GM-CSF, which is an Amgen drug. And back in, I think it was the early 90s, mid 90s, they started seeing some adverse effect in patients. And that was when the people started developing antibodies against, actually, I think it's GCSF, Neupogen, that they started developing antibodies against Neupogen. And that is where the immunogenicity field kind of came out of that. And that's why it's on the label now. It's a big part of the bioanalytical development of a drug. And it's a huge part of the warning box, the black box, as we've talked about. But really, what was happening was people were misstoring the drug. Some people it was cold and they were microwaving it. Well, you can imagine microwaving a protein, John, is going to cause you to generate an antibody against it. So, I mean, this whole immunogenicity field might have came out of an era, and it is a very expensive field. Now, I think it's good. Of course, it's led to neutralizing antibodies, and we don't need to talk too much about that. But John, I think what's happening now is we're getting long in our podcast here, and sometimes I sit and say, what are we going to talk about? And man, has this gone by very fast, John. We could sit here and talk a lot longer about these subjects. We didn't even talk about some of the challenges with oncology and orphan drugs, but let's maybe take one quick second to talk about that in the clinical setting, meaning the length of time of an oncology trial becomes very challenging on the biologic side or the large molecule side. Most notably is reagents. That managing those reagents over the life of an oncology trial can be very, very hard. And what I mean by that is the capture antibody is frequently some sort of anti-id antibody that you've made. And normally when you make that, maybe you can get a large supply that'll last you. So that's the least of your worries. But then in the ADA setting, you frequently biotinylate and ruthenium label reagents to do your bridging ADA assay. Now you've got two more critical reagents to manage. And then, of course, any sort of biomarker assays, you want to try and keep the same lots of those reagents. And those typically only have a shelf life of like a year. So that becomes a major thing to manage in the oncology space on the biologic side. And then also orphan drugs, I think, fall into a challenging category just because those trials can go on for years at a time and only accrue maybe 5, 10, 20 patients and still get approval off a very low end number. So, John, I know I'm bashing through things there, but I'll take a breath and give you a second to comment on that.
Yeah, oncology obviously is a hugely important field in terms of trying to address, what is a very nasty disease on so many different tissues, etc. But yeah, for us bioanalytically, oncology can be a nightmare. With every project that I'm talking small molecule again here. Every project you've got development validation, then you're geared up to do samples, and we're always planning ahead. So forecasting is a big part of what we do. So we figure out, do we have the capacity? If we don't have the capacity, how do we gear up, etc. If you're trying to forecast on the basis of oncology studies, it's really, really tough. Like you say, studies can go on a very long time. if in the dose escalation portion of studies, they can be tiny. What you don't have is a real handle of how quickly it's going to escalate. So for a typical dose escalation study out of, say, a cardiovascular drug, you would expect the study would start on a certain day. The first cohort would be, say, 12 to 20 patients, would say 12 samples each. Those would be shipped out after a couple of days to the bioanalytical lab for the assay. You'd run the samples, feed the data back, and then the decision would make whether to go forward to the next escalation dose level. Same deal again, 12 to 20 patients, 12 samples, run the samples, feed the data back, ready for the next dose the week after that. That's a fairly typical time frame, and these studies maybe last 8 to 10 weeks. In oncology, you'll start, maybe your first cohort will be 3 patients, but it depends on the being able to recruit someone with that disease condition that still meets the criteria of the clinical protocol to even allow them into the study. You might get 1 patient, but then you're looking around and it takes a while to recruit the second and then the third, and that's just your first cohort. You've only got maybe, say you get 10 samples per patient, you send those samples out, you still want your data really, really quickly, so for the bioanalytical lab, suddenly these samples might turn up out of the blue and you're not really that prepared for it. And then it's sit tight for however many months, because you might have a second cohort, but again, in terms of recruiting, it can be a real challenge. So in terms of logistics on the bioanalytical lab, becomes really, really tough. Although we know it's really important and we want to be supporting oncology studies, it's one of those things where you definitely see and think, oh, not another oncology drug, because you know how it can wreak havoc with your scheduling, etc.
Yeah, John, we support a stem cell therapy. It's gene therapy. It falls into that category. And we support their ADA program on top of it's an enzyme replacement type of, well, not replacement. It generates its stem cell treatment to make an enzyme that is lacking. And that's the orphan status. Well, we have a 7-year contract to test like probably less than 20 total samples or 20 patients due to the rarity of the disease. But we're in year 2 of that 7-year contract, and we've run 3 batches of ADA. Like you said, it wreaks havoc on managing, and it's quite an investment by the sponsor. But in the same token, the assay is still very sensitive, and we've really helped with treatment of these poor children. This is in children. I think, John, this is almost its own topic. I feel like we could sit here for another hour and talk about the challenges of oncology and orphan drugs. But and even like the last item on our agenda was large molecules and specifically ADAs and NAbs. And we're going to press pause there, John. And what we'll do is, if you ever go on vacation, which I hope you get one soon, I'll bring in a guest host and we'll make that the topic. Okay. I think for the sake of time, because we are long, John. And by the way, both of us are pretty fired up, because this is striking. I can feel it coming out of you. This is striking a chord in you, certainly in me. I'm getting like, wow, there is a lot of things we manage. Sometimes when you're in the weeds doing it, you don't think about it. But as you pull back, like we did today, it's like, dang, we really do quite a bit of management of a number of things that I think a lot of companies probably don't even think about. They don't realize what they have ahead of them in terms of trying to manage their bioanalytical drug development assays. So with that, John, let's press pause, unless you have one last thought. Go ahead.
One last thought. Yeah, so we did it. We even had one more topic within this, and that's in terms of sample logistics. And it's particularly touching on larger studies and central labs and overseas shipments, and we'll hold that to another time. And actually, we also, one thing I want to touch on was working on studies that then go into China, because that actually brings a lot of very interesting challenges. But I think we know this is a series. Let's see if we can slot this in another podcast, because it's a really fascinating, and it's sort of fun to talk through what you dealt with and the practical approaches, etc. But yes, let's move on.
So we're being told that this week we had no feedback and questions from our audience. We're always looking for any sort of feedback or topics you'd like us to discuss. So please don't be bashful out there. Go ahead and let us know, either on our YouTube page or by Twitter or even by LinkedIn. But we really appreciate your feedback, and we want you to let us know how we're doing so that we can constantly improve the podcast for you.
Exactly. We'd love to Yeah, any feedback is welcome, and we certainly want to assimilate comments into what we're doing, particularly if it's new topics. So at this point, we'll wrap up. So as we've mentioned, we're in the middle of a series of four um, podcasts, all covering where does bioanalytical fit in the drug development process. This we've just completed part two, and obviously I think we could probably go beyond four if we keep talking like this. However, next week is we have a holiday weekend due to Independence Day, so we're thinking we'll probably take a break from this series and pick it up the week after. So what one thing we thought about, we'd step away and maybe talk about how does a CRO handle holidays like Independence Day, Thanksgiving, Christmas, and any type of break like that, because it's in our interest as much as possible to keep business going.
Yeah, John, I'm looking forward to pressing pause on our series of um, bioanalytical and how it fits into the drug development process, and excited to talk about some of the challenges that are we experience as a CRO during the holiday timeframe, because we are a 24/7, 365 organization. But I won't tip our hand. There's lots of things and ways we manage that, but certainly should be quite an interesting topic around, kind of maybe behind the scenes for those of you that aren't that familiar with the CRO space, just how we manage all of that. Where we never stop working is our motto, John. That'll do it for this week's podcast.
Yeah, absolutely. I think on that topic, I very much have enjoyed this and look forward to doing it in a week's time. Thank you very much.